Yes, today we are going to start with the circulatory system, a very important system for our body, for the nutrition of the different body tissues as well as the organs. So circulatory system, as this is a system, so it comprises of different types of units. But first we have to understand two different types of circulatory system. That is open circulatory system and closed circulatory system. Circulatory system. Open circulatory system and closed circulatory system. Open circulatory system comprises of open channels. That is the circulating fluid. The circulating fluid is not transporting inside any vessel. That is why it is known as open circulatory system, and these open spaces are known as lacunae. So these open channels are known as lacunae. So these lacunae have these circulatory fluids. So as these are not flowing in any vessel, so the lymph as well as the blood is mixed. That is why the circulating fluid is known as hemolymph. The circulatory fluid is known as hemolymph. The open circulatory system comprising of open channels called lacunae, in which this circulating fluid hemolymph flows. Why it is called hemolymph? Because it is not in the mixed, it is in the mixed part, that is the heme, that is blood, and the lymph are mixed. That is why it is known as hemolymph. And in this type of open system, open circulatory system, the hemolymph baths over the organ. It baths the organ and the tissues to supply oxygen and nutrition. That is why it is called open channel. As this is an open channel circulating fluid, that is why the pressure is very low. The pressure is low as compared to the closed circulatory system. So this open circulatory system usually found in arthropods and some annelids. So what is open circulatory system? Open circulatory system comprises of open channels through which the circulating fluid is transported, and due to the open channel the pressure is very low. Now these are found in arthropods and annelids. Now coming on our closed circulatory system. Closed circulatory system. Closed circulatory system. So in closed circulatory system, this comprises of blood vessels and heart, the pumping organ. So in this the blood and the lymph flows into the vessels. That is why it is known as closed circulatory system. As compared to the open circulatory system, the pressure in the vessels is much more than the lacunae. So this comprises of blood vessels and the heart. Now we are going to deal with the blood. What is blood? The circulation of the fluid that is called blood. So blood is what? It is a fluid connective tissue. Why it is called fluid connective tissue? Because it has the property to flow. It has the property to flow. Now coming on the composition, what will be the composition? So it comprises of a liquid part and the cellular part. Liquid part is known as plasma and cellular part is called hematocrit volume. It is called hematocrit volume. The liquid part is called plasma and the cellular part is called hematocrit volume. Now, this liquid part comprises of about 50 to 55% of blood and its 95% is water and contains different types of vitamins, minerals and proteins. Vitamins, minerals and proteins. So this part is plasma, that is the blood minus cellular part or the cells is known as plasma, the liquid part in which the cellular component is mixed. So it is 50 to 55% blood, 95% is water, vitamin, minerals and proteins. So here, apart from all the components, there are important 3 proteins are found. Important proteins, that is globulin, albumin and fibrinogen. Globulin, albumin and fibrinogen. These are the 3 main proteins found in the plasma, which is very helpful in the body defense, in the osmotic pressure maintenance and in the clotting of the blood. This globulin helps in the immune system. That is, it is basically Ig, immunoglobulins. So it resists the foreign agent to enter inside the body. Albumin, it maintains osmotic balance of the blood, so that there will be no plasmolysis or no bursting of the cellular part takes place. And fibrinogen helps in blood clotting. So these 3 proteins are very helpful for the body. Globulin helps in immune system, albumin maintains osmotic balance of the body and fibrinogen helps in blood clotting. So now coming on the cellular part. There are majorly, it is divided into 3 parts, that is RBC, then we have WBC, then we have platelets. RBC, WBC and platelets. RBCs are erythrocytes, WBCs are leukocytes and platelets are thrombocytes. RBCs, WBCs and platelets. erythrocytes, leukocytes and thrombocytes. Now we are going to deal RBCs separately. Now this WBC will again divide it into granulocytes and agranulocytes. Granulocytes and agranulocytes. Granulocytes will again divide it into three parts, that is acidophils, neutrophils, basophils. Acidophils. And agranulocytes are lymphocytes and monocytes. Lymphocytes and monocytes. Now we are going to deal with the RBC first. Now first we are going to deal with the RBC, that is red blood cells or red blood corpuscles. Red blood corpuscles. So these are red in color. And the process of formation of RBC is erythropoiesis. The formation of RBC is known as erythropoiesis. Now second point about the RBC, the site of formation. The site of formation of RBC in adult and in child. In adult it is only bone marrow. It is only bone marrow, and in child it is bone marrow as well as liver. Liver is also producing RBCs in children. Okay. Now, third thing about the RBC, the shape of the RBC. What will be the shape of the RBC? Shape means when it is immature or when it is formed and when it is mature. When it is immature, it is spherical or oval because nucleus is present. But in mature condition, it is biconcave because the nucleus becomes degenerated. That is why it becomes biconcave. So the process of formation of RBC is erythropoiesis. The site of formation in adults is bone marrow, but in children this is bone marrow as well as liver. In the shape, as far as the shape is concerned, the immature becomes the spherical or oval and mature is biconcave due to degeneration of the RBC. But in the case of llama and camel, nucleus is present in the mature RBC. So So this exception should be in the mind. Now next point about the RBC is that it is red blood cell. Why it is red? Because of a pigment called hemoglobin. Because of the pigment hemoglobin. This is red in color. So it is a conjugated protein. The chemical nature is it is protein. It is not carbohydrate. It is not any other fat. Conjugated protein. Because it is having two parts. If we split it, it is having a protein part and a non-protein part. This is heme and it is the protein part. Globin is the protein part. Heme is the base metal is iron and this is the protein. So in this the protein is associated with the iron. That is why it is known as conjugated protein. Now heme in the iron is in the +2 oxidation state, that is in the ferrous state in the hemoglobin, and the main function of hemoglobin is to transport respiratory gases. Respiratory gases means oxygen as well as carbon dioxide. So here one thing should be in the mind that RBC not only transports oxygen but also carbon dioxide. Now here when hemoglobin forms bond with the oxygen, it is irreversible. So it is called, the product is oxyhemoglobin. When it forms with the carbon dioxide, it is called carboxyhemoglobin. And with the it is forming oxyhemoglobin with the carbon oxygen, and with the carbon dioxide it forms carboaminohemoglobin, and with the CO it forms carboxyhemoglobin, and with the methane it forms methanoglobin. But one thing should be in the mind that this is reversible. This is reversible, but this is irreversible. So hemoglobin binds with the carbon monoxide 200 times than of the oxygen or carbon dioxide, but this is irreversible bond. So hemoglobin becomes less available when there is carbon monoxide poisoning takes place. So hemoglobin, the red pigment because of which RBC is red in color, is a conjugated protein, and it transports oxygen as well as carbon dioxide, and one hemoglobin molecule transports 4 oxygen molecules, or we can say 4 oxygen molecules or 8 oxygen atoms. So the bond between hemoglobin and oxygen or hemoglobin and carbon dioxide is reversible, but it is of dative type. It is not covalent bond, it is not the electrovalent bond, it is a dative bond or coordinate bond. So this hemoglobin provides the transportation, and the amount of hemoglobin is about 12 to 16 gram per 100 ml of blood. per 100 ml of blood, it is about approximately 12 to 16 grams. Okay. Now, the RBCs are manufactured or produced in the bone marrow as well as in the liver in children's, but these are having 120 days approximately lifetime, and they are getting destroyed and submitted to spleen for further processing. So, in the case of more than or abnormal increase in the RBC destruction, there will be swelling of the spleen takes place, as in the case of malaria. So that is known as spleen index. So spleen index is seen in the case of malaria. So this is about the RBCs. The amount of RBCs ranges from 1 to 5.5 to 5.5 million per millimeter cube of blood. Per millimeter cube of blood. So RBCs, but above all RBCs, among all the cells, RBCs are in largest number. So coming on the our next type of cell, that is WBC. That is WBC, white blood corpuscles or leukocytes. They ranges from 6,000 to 8,000 per millimeter cube of blood. It is lesser than the RBC, but these are equally or more important for the resistance power of the body. And these are of two types, that is, as we have earlier said, that these are granulocytes and agranulocytes. Granulocytes means neutrophils, those having granules in their cytoplasm, neutrophils, basophils and acidophils or eosinophils. This and this are phagocytic. These are also phagocytic, and their number increases during the allergy. Basophil produces three types of chemical substances, that is histamine, serotonin, which helps in the allergic reactions, and heparin. It is an anticoagulant. Heparin is an anticoagulant. So histamine, serotonin and heparin is produced by the basophils, and neutrophils and acidophils are phagocytic in nature. That is, they will engulf the material or the foreign material which can cause disease. So these are granulocytes, that is those cells or those WBCs which have granules in their cytoplasm. Now agranulocytes, what is agranulocytes? These are of two types, that is lymphocytes and monocytes. Lymphocytes and monocytes. Monocytes are what? Monocytes are phagocytic, largest phagocytic cells. That is, they will engulf the foreign agents And lymphocytes are of two types, B lymphocytes and T lymphocytes. This is producing antibodies and this is producing perforins to kill the foreign agent or the pathogen which can cause disease. Lymphocytes and monocytes are agranulocytes. The whole RBC, WBC as well as the platelets comprises of hematocrit volume. Now coming on our next cell, that is thrombocytes. Thrombocytes or platelets, blood platelets. Thrombocytes or blood platelets. So these are also produced in bone marrow, but from specialized cells known as megakaryocytes. Produced in bone marrow from megakaryocyte. So these can amount from 1.5 to 3.5 lakhs per 100 or per millimeter cube of blood, and these helps in the main function of the platelets is helps in blood coagulation. In the absence of it, there will be the death of the person if blood do not clot. So now we will see how platelets helps in the clotting of the blood. So now the process of blood coagulation. The process of blood coagulation. Now see, there is an injury. When there is an injury, at the site of injury, the platelets as well as the WBCs rushes at the site of injury and produces a chemical substance known as thromboplastin. And the rushing of the WBCs as well as the platelets at the site of injury is known as diapedesis. So at the site of injury, thromboplastin is released by the WBCs as well as the platelets, and this thromboplastin will help in the conversion of prothrombin to thrombin. This is inactive protein found in the plasma, and this is the active protein found in the plasma. Okay, so this thromboplastin activates as catalyst for the conversion of prothrombin into thrombin in the presence of calcium ion. What? In the presence of calcium ion. And this thrombin will convert the soluble fibrinogen into fibrin. This is soluble in plasma and this is insoluble. And this will form mesh-like structure and traps blood cells and forms the clot. It will form the clot. So it will form a mesh-like structure and traps the blood cells and forms the clot. So this time from injury to formation of clot is known as clotting time. Is known as clotting time. This is known as clotting time. So this clotting time can vary from 2 to 8 minutes. And if there is increase in the clotting time, then there must be some deficiency or there must be some disease that can be or that should be cured. Otherwise, it can cause deadly disease or result in the condition that is known as anemia. So, this was the process of blood coagulation. Now, some of the substances which prevent blood coagulation are known as anticoagulants. Anticoagulants. So, anticoagulants of animal origin can be anophyllin and hirudin from hirudinaria. Anophyllin from Anopheles mosquito, and sodium or potassium citrate or oxalate are mixed in the blood to prevent clotting of the blood for the purpose of storage in blood banks. What it does, it prevents clotting of the blood by the precipitation of calcium ion. So when calcium ions are precipitated out, then there will be no conversion of the prothrombin into thrombin, and as a result no further step will be there for the coagulation of blood. Now these are all about the anticoagulants. So this type is blood is very necessary for the body because it transports respiratory gases as well as different types of vitamins and nutrition or minerals. Now there are some conditions such as polycythemia. Polycythemia, that is abnormal increase in RBC. When there is abnormal increase in RBC, that condition is known as polycythemia. And when there is abnormal decrease in RBC count, then it is known as anemia. Anemia. So anemia is not a disease. It is a condition in which there is abnormal decrease in the RBC number. So anemia can be of many types: nutritional anemia, megaloblastic anemia, it can be pernicious anemia, and it can be thalassemia and sickle cell anemia. Sickle cell anemia. Nutritional anemia, megaloblastic anemia, pernicious anemia, thalassemia and sickle cell anemia. So this is the condition when there is abnormal decrease in the number of RBCs. Polycythemia is the abnormal increase in RBC number. Now this is about the blood. Now coming over our blood vessel. Next component of the closed vascular system is blood vessels. What is this? Blood vessels. So blood vessels of many types, but important ones are arteries, arterioles, capillaries, venules and veins. Arteries, arterioles, capillaries, venules and veins. Now generally arteries contains oxygenated blood and veins contains deoxygenated blood except pulmonary artery and pulmonary vein. The pulmonary vein contains oxygenated blood and the pulmonary artery contains deoxygenated blood. Now blood vessels, arteries and veins, the layers found in arteries and veins. There are three layers in arteries and veins: tunica, tunica externa, tunica media and tunica interna. Tunica externa, tunica media and tunica interna. These are the three layers found in the blood vessels. But in the case of arteries, the lumen, this space is known as lumen. The lumen is lesser than the veins. The vein have wide lumen as compared to the arteries. So tunica externa, tunica media and tunica interna. So these are the blood vessels. Arteries transports oxygenated blood and veins transports deoxygenated blood except pulmonary arteries which transports deoxygenated blood. So the main criteria of arteries and vein is not the transportation of oxygenated or deoxygenated blood. As far as the organ is concerned, say this is an organ. The vessels which transports towards the organ, that are arteries, and which transports blood away from the organ, that are known as veins. As far as the organ, any organ concerned except heart. In the case of heart, this criteria will be opposite. So the criteria of deoxygenated and oxygenated blood is not perfect for the arteries and the veins. Now coming on our pumping organ, that is heart, without which the circulation is not possible. And if circulation is not possible, then there might be the death of the tissues as well as of the person can take place. Now let's have a look on the mammalian heart. It comprises of just an indicative diagram. It comprises of two atria and two ventricles. This is left atria, this is left ventricle, this is right atria, right ventricle, this is interauricular, septum. This is interventricular septum. Interventricular septum. Now these are valves, bicuspid and tricuspid valves. These are bicuspid valve. These are tricuspid valve. And these valves are attached with the myocardium, that is with the help of strings known as chordae tendineae. Chordae tendineae. These are also to prevent reversing back of the valves when there is contraction of the ventricle takes place. This is the general parts of a mammalian heart. So, two auricles and two ventricles. Upper chambers are known as atria, lower chambers are known as ventricles. It is divided by interventricular septum and interauricular septum. Now, this mammalian heart, in the case of humans, it is present in the space between the lungs, that is known as mediastinal cavity. Mediastinal cavity. The cavity is known as mediastinal cavity between the lungs. So, heart is present in the cavity between the lungs known as mediastinal cavity and covered by a membrane called pericardium. So it is covered by the pericardium. Now, this is having, the heart is having nodal tissues. Nodal tissues. As it is a myogenic heart, the mammalian heart is myogenic heart. Why it is called myogenic heart? Because the action potential is generated from nodal tissues found in the cardiac muscles. So these are of many types, such as SA node, AV node, bundle of His, and Purkinje fibers. This is AV node, this is bundle of His, and these are Purkinje fibers. Nodal tissues. The action potential, that is the heartbeat, starts from the or originates from the SA node. That is why it is also known as pacemaker, and this is pace setter. AV node is pace setter, that it will maintain the impulse which is going towards the ventricles. So the conduction of the action potential takes place from SA node to AV node, SA node to AV node, to bundle of His, and to Purkinje fibers. So the impulse conduction way is SA node to AV node to bundle of His to Purkinje fibers takes place. Now, as far as the conduction of impulse takes place or the action potential takes place, this will be the path. But now, what is cardiac cycle? So heart is simply a pumping organ. So this pumping organ is going to pump the blood, and those cyclical changes which takes place during the pumping of the blood by the heart is known as cardiac cycle. That is cardiac cycle or heart beat. So this cardiac cycle comprises of systole, diastole and joint diastole. Systole means contraction, diastole means relaxation, and joint diastole means relaxing of all the PAM chambers, of all chambers. So basically the filling of the chambers takes place at joint diastole,